Hello, I'm Pierre Marrec, and I'm going to present you my work about a predictive understanding of the planktonic food web on the northeast US shelf, with a focus on the new insights from interannual variability and disturbance. This work is part of the NES-LTER project and was done with the collaboration of Diana Fontaine, Tatiana Reinerson, and Susanne Menden-Doyle. Let's start with the planktonic food web and the importance of microzooplankton grazing. Microzooplankton are protist cells smaller than 200 micron, which consume up to 70% of daily net primary production in the global ocean. Microzooplankton are a crucial link between primary producer and higher trophic levels. The goal of this work is to get a predictive understanding of the planktonic food web. For this, we aim at identifying the biotic and abiotic factors controlling phytoplankton growth and microzooplankton grazing. It's particularly important because phytoplankton grazing by microzooplankton is still poorly constrained, especially in dynamic and changing coastal oceans. Our coastal ocean study area is the Northeast US Shelf. In 2019, the Northeast US Shelf long-term ecological research site was established in this ecologically and economically important ecosystem, where polar cold water from the Arctic flows south along the coast, while warmer waters are carried northwards by the Gulf Stream, as you can see on the satellite sea surface images. This ecosystem is highly productive, as shown by the high chlorophyll concentration observed on the shelf, and is characterized by intense seasonality with an amplitude in terms of temperature up to 5 degrees between winter and summer. In addition, the NES ecosystem is subject to significant physical and ecological alterations in the context of global change. Our study area is represented in red here. We have here highly contrasted environmental conditions in a limited area. We can describe this condition as a sharp gradient between polar-like conditions to tropical-like conditions, from highly productive coastal water to oligotrophic Gulf Stream waters. This is thus an ideal site to investigate how the planktonic food web changes through space and time in response to changes in the physical environment, and how those changes impact ecosystem productivity, particularly of higher trophic levels. This map represents our study area on the ship track from the last summer cruise from Martha's Vineyard to the shelf break, performed on board the Endeavour. Since 2018, we've performed 4 winter and 4 summer cruises along the transect. During each cruise, we sampled 4 to 7 surface stations, and we performed 24-hour on-deck incubation experiments. We are using the 2-point dilution method to measure phytoplankton growth rates and microzooplankton grazing rates based on chlorophyll budgets. Since 2019, we've measured simultaneously C13 net primary production. In the next few slides, I'm going to present the seasonal and cross-shelf patterns observed along the transect. A first description of these patterns was made in Marrec et al. 2021. In this study, we added 2 winters and 3 summer cruises, which support remarkably well our first description. Let's start by the sea surface temperature and salinity observed in the 4 hydrographical provinces from near shore to off-shelf waters. We observe both in winter and summer colder and less salty waters in coastal regions than in offshore ones, with consistent patterns during our 4-year studies. This figure represents size-fractionated surface chlorophyll concentrations along the transect, from the shore to the shelf break, in winter in blue and in summer in red. In light blue and red is represented the contribution of large phytoplankton cells. Winter is characterized by high surface chlorophyll concentrations dominated by large shells, while in summer we observe lower chlorophyll concentration with the phytoplankton community dominated by small cells. We observe in both seasons a decrease in terms of chlorophylla from the shore to the shelf break, with a decreasing contribution of large phytoplankton cells. Phytoplankton growth rates expressed in per day indicate that phytoplankton grow faster in summer in red than in winter, and in offshore than in coastal waters. When looking at microzooplankton grazing rates, also expressed in per day, we observe that grazing rates are relatively similar across seasons and cross shelf. In winter, grazing rates are in the same range as phytoplankton growth rates, while in summer, growth rates are much higher than microzooplankton grazing rates. We use the ratio of grazing divided by growth to estimate the percentage of primary production consumed by microzooplankton. These percentages are represented along the shelf on this figure. As suggested by the growth and grazing values in the same range observed during winter, the percentage of primary production consumed by microzooplankton in winter is close to 100% in coastal water and decreased down to 50% in offshore waters. We have a direct and efficient transfer of organic matter from phytoplankton to the microzooplankton in winter, suggesting simple trophic interaction in the planktonic food web. While in summer, the percentage of primary production consumed by the microzooplankton never exceeds 50%, which indicates a reduced role for microzooplankton as conduit of carbon in the planktonic food web. These low values suggest more complex trophic interaction in the planktonic food web, such as trophic cascade or mixotrophy. As mentioned earlier, C13 net primary production measurements were performed simultaneously as our gross grazing rates measurement. Phytoplankton growth can be converted into primary production based on carbon to chlorophyll ratio. We observe a significant correlation between the primary production estimated from growth rate and the C13 net primary production, which allow us to express phytoplankton growth and microzooplankton grazing in carbon fluxes. I represented here the grazing loss expressed in microgram per meter cubic per day in winter and in summer along the shelf. For the first time, we were able to quantify the amount of carbon transfer from phytoplankton to microzooplankton on the northeast shelf, which is an essential metric to better understand the food web dynamics. I showed you that we observe consistent seasonal and latitudinal patterns in biological production rates on the shelf. Another way of representing the seasonal mismatch between phytoplankton growth and microzooplankton grazing rates is to plot them like this, with growth rates on the x-axis and grazing rates on the y-axis. This representation clearly shows the decoupling between phytoplankton growth and microzooplankton grazing during summer, which reduce the trophic transfer efficiency in the planktonic food web at this time of the year. In the second part of this talk, we are going to focus on some observed deviation from these seasonal patterns. So let's see some new insights in terms of planktonic food web function and structure we can get from interannual variability and disturbance. By looking at the seasonal growth versus grazing rates, we can see that in winter patterns, here coupling between phytoplankton growth and microzooplankton grazing, are sometimes observed in summer. In summer 2019, as represented by the magenta circles, we observe high trophic transfer efficiencies, same as during winter. I represented here a principal component analysis representing all the sample stations based on temperature, salinity, light availability, chlorophyll concentration, percentage of small phytoplankton cells, growth, and grazing rates. We can observe a clear separation between summer and winter stations and between coastal and offshore ones. The two summer stations of interest are the one appearing in the winter half of the PCA space, showing similarity with winter stations. When looking at the temperature, salinity, chlorophyll concentration, and percentage of cells smaller than 10 micrometers, during the 4 summer cruises in nearshore and mid-shelf waters, our stations were characterized by high temperature and low salinity compared to the other years. More remarkably, in summer 2019, we were in presence of an exceptional and intense bloom of large diatom cells. You can find additional information about this exceptional summer bloom of diatoms in Alejandra Castillo poster describing the anomalies in terms of primary production observed during this bloom. This disturbance from the classic summer seasonal pattern highlights the importance of the phytoplankton community structure. But as you can see on the left plot, similar coupling between growth and grazing rates was observed in summer 2021, represented by the yellow dot. At this time, the temperatures were relatively low compared to other years with relatively high salinity. And we were in presence of a classic summer phytoplankton community structure characterized by low chlorophyll concentration and with small cells dominating. This exception in terms of deviation from the consistent seasonal pattern suggests that other biotic characteristics might be driving the flow of carbon through the planktonic food web. This other biotic characteristic could be the structure and the composition of the microzooplankton community, a component that we are going to study more in details in the future. I could tell you a reverse summer in winter story, but I don't have time for this today, unfortunately. Briefly, you can see that low or lack of grazing are sometimes observed in winter, and these 2 stations clearly show up in the summer half of the PCA reduced space. The main deviation in terms of abiotic and biotic factors was the phytoplankton community structure. I will tell you the full story in a paper in preparation. To conclude these little investigations, we observed that deviation in grazing intensity co-occurred with deviation in temperature, salinity, but most remarkably with chlorophyll concentration on the phytoplankton community structure. Abiotic factors are often limited to fully explain deviation in grazing, and the phytoplankton community structure is an essential biotic parameter to consider. Along this presentation, we first characterize consistent seasonal and latitudinal patterns in biological production rate. For the first time, we were able to quantify the flow of carbon from primary producers to microzooplankton grazer on the northeast shelf. Then, we showed how interannual variations and disturbance can separate the influence of biotic from abiotic drivers in terms of trophic transfer efficiency, and that decoupling of typical conditions among biotic and abiotic factors can be used to identify specific single or multiple drivers. Ultimately, we showed that phytoplankton community structure rather than abiotic factors control trophic coupling in the planktonic food web. The 2 main information to keep from this talk are that we made considerable progresses to predict seasonal fluctuation in planktonic food web structure and function in the northeast shelf. Such predictive power is essential to better understand how disturbance and climate change might affect carbon flow from the planktonic food web to the higher trophic levels in the future. After this, I would like to acknowledge all the PIs on the NES-LTER project and all the NES-LTER team, and of course the captain and crew of the best research vessel ever, the Endeavour, and also the Marine Tech on board. Finally, I would like to thank all the present and past Mendon Lab members who contributed to this work. Thanks for your attention.